Good afternoon. I am Dr. Neeraj Mishra, Assistant Professor in the Department of Pediatrics, and today we will discuss disorders of sexual differentiation. In this class, we will try to see how the sexual organs are developed, what is the hormonal influence, and what are the disorders related to the development of sexual organs and related to the deficiency of hormones, including congenital adrenal hyperplasia. So DSD is defined as congenital condition in which there is a discrepancy between chromosomal, gonadal, and anatomical sex. Whenever you are seeing discrepancy, if a person is looking like male and his or her chromosome composition is female, like 46XY, XX is female, and he is looking like male, then it's a disorder of sexual differentiation. If you are trying to see her genitalia, and you are not, by seeing the genitalia of the baby, you are not able to make out either this genitalia is male or genitalia is female, then you can say that it is DSD, disorders of sexual differentiation. So for the clinical purpose, this disorders of sexual differentiation has been classified into three types: 46XY, 46XX, and sex chromosomal disorder. So before going into the detail of this DSD, we should know how the genitalia are get developed from the embryo. So the gonads are bipotential till 7th week of life. Means, but having said that, the male and female differentiation of gonads takes place after 7th week of intrauterine life. This male and female differentiation starts at 7th week. Till 9th week of age, you are not able to differentiate, although differentiation has started, but you are not able to differentiate that this embryo is male or female. After 9 weeks, this differentiation is almost complete, and you can make it out that it is male and female. The presence of Y chromosome, and on the Y chromosome, there is a SRY gene, sex determination region of Y chromosome, that is called SRY gene, compels the embryo to develop into male, and by convention, this female differentiation is a default setting. When this signal from Y chromosome, from SRY gene, is absent, the embryo will develop into female. It is the conventional teaching that default setting of the embryo is to develop into female. But recently, some genes and differentiation factors have been identified, which said that apart from absence of SRY gene signal, some other signaling pathway also exists, which transforms the bipotential embryo into female gonads. So this should be, I want to make it clear. Okay. The presence of more than one X chromosome does not alter the initial development of the embryo. Having said that, till 13 to 15 weeks of intrauterine life, even in the absence of one X chromosome or in the presence of one extra X chromosome, the fate of that gonad is not going to alter. But as soon as the primordial follicles arise around 13 to 15 weeks of intrauterine life, this gonadal failure sets in. And for example, in the Klinefelter syndrome 47XXY, one extra dose of X chromosome is there, and after 10 to 15 weeks, this meiotic failure will take place, and ultimately the gonad will be the strict gonads. But in both the cases, either the male or female, till 13 to 15 weeks of life, this gonadal failure will not set in. If chromosome composition is 47XXY, means XY disorders of DSD, there is a high risk of development of gonadal tumors, especially gonadoblastoma. So, if the baby will not be a fertile baby, gonadectomy, early gonadectomy is advised. As I said, early development of ovary does not require 2X chromosome, but after 15 weeks, ovarian development fails, resulting in a stick gonads characteristic of gonadal dysgenesis syndrome. So, see, this is the bipotential. First image is a bipotential gonad, where you can see both mesonephric and Wolffian duct. Okay, mesonephric duct, Mullerian duct, and Wolffian duct. Mullerian duct is the structure from which female internal genitalia develop, and Wolffian duct is the structure from which male internal genitalia develop. As you can see, before till 7th week, both Mullerian duct and Wolffian duct is present in the embryo. But with the signal from SRY gene, this Wolffian duct will develop into epididymis, testis, vas deferens, seminal vesicle. Okay, and this SRY gene signaling pathway induce the leading cells to secrete testosterone. At the same time, from Sertoli cell is secreted MIS, Mullerian inhibiting substance, or also called AMH, anti-Mullerian hormone. This hormone from Sertoli cell will induce the regression of Mullerian duct, so that this embryo will not develop into the female. So females are suppressed right from the embryonic life. Although I told you default setting of the embryo is to develop into female, but due to inhibition by this MIS or AMH, this female development is suppressed, and under the presence of SRY gene signaling, this Wolffian duct will lead to the male genital structures. So if signal from the SRY gene is absent, this Mullerian duct will develop into ovary, fallopian tube, uterus, and upper part of the vagina. Upper 2/3, you can say. Lower 1/3 of the vagina is derived from urogenital sinus. You may be knowing from your anatomy, embryology knowledge. So external genital development is under the hormonal influence, and it develops from 9 to 14 weeks under the influence of AMH, testosterone, and dihydrotestosterone. Female ovary in the embryonic period as such is quiescent, but it has been postulated that some aromatic activity is there which produces estrogen from the peripheral converter. So what should be the clinical approach to the infant with suspected disorder of sex development? So as you know, first we should know when we can say that this baby is ambiguous, and more correctly nowadays term has been used as DSD, disorders of sexual differentiation. So what are the criteria when you will say that this baby has DSD? So this slide has been taken from Guwahati 8th edition. So there are certain soft markers they have given that you can say that this baby has DSD. Like by naked eye examination, you are not able to differentiate the genitalia. You are not able to make out either it is male or female. So you can say that it's ambiguous genitalia. A penis and bilateral non-palpable testis. Penis is there, but testis is not palpable. Then you can say that it is DSD. Because this penis, you are not sure why we are keeping this penis, this bilateral non-palpable testis as DSD. Because by seeing the penis, what it looks to you is penis maybe hypertrophic clitoris, and what you are seeing as testis maybe labia majora, okay, and labioescrotal folds. In the same way, unilateral cryptorchidism with hypospadias falls into the category of DSD. Any severe degree of hypospadias like penoescrotal, scrotal, and perineal hypospadias with microphallus or without microphallus with undescended testis falls in the category of DSD. Apparently, female appearance with enlarged clitoris or inguinal hernia, asymmetry of size, pigmentation, or location of labioescrotal tools, discordance of external genitalia with genital cavity, all these falls into the category of DSD. So, if you are going to see abnormal genitalia, you should know what is normal genitalia. So vaginal opening, fully visible, 3 to 4 millimeter slit like orifice, clitoris 22 to 6 millimeter. More than 6 millimeter clitoral length is called clitoromegaly. Absence of gonads in labia, Mishra or Gonadal. Male baby, the penis size should be 2.5 and bilateral testis normal in size and position. In history, the mother will not give you any history of clear-cut history of DSD, but there are some pointers in the history by which you can make out that this family is suffering from DSD or this baby is suffering from DSD, like history of infertility in the family, amenorrhea in the family, hypospadias or infants with unexplained death in the family, history of virilization of both mother and the baby, history of parental consanguinity drug intake, and artificial reproductive technology are the soft markers, soft pointers, which will tell you that this baby is suffering, this baby might have DSD. Physical examination you have to do for both the male and female baby, and general signs of dysmorphism, features of hyperpigmentation, associated anomalies including skeletal malformations. External genitalia you have to look for, and more common Prader classification commonly used to stage realization of female external genitalia and external muscularization is called EMSA, great male muscularization. So you should be aware of this Prader classification and external EMS score. So here you can see no virilization, a stage 1, some fusion, stage 2, stage 3, and stage 4, stage 5. So stage 1 is normal female genitalia with clitoromegaly. Stage 2 partial posterior labial fusion with clitoromegaly. Stage 3 labioscular fusion so that there is a single opening in the perineum and clitoromegaly. Fusion of labioscular fold so that a single opening is at the base of the phallic structure, appearance like a male external genitalia. This is actually female, but the male external posterior labial fusion is very high degree, and clitoris is totally enlarged, so it will look like a male genitalia. Same in case of EMS score. You can see scrotal fusion no, S. S it will score 3. Micropenis no, then it will score 3. Urethral meatus normal, score 3. Right gonads. So where is the gonad? Abdomen, gonadal folds. So by this way you can use scoring. Any score less than 11 is indicative of DSD. This is the penis and different variety of hypospadias. So perineal, scrotal, and penoscrotal hypospadias is considered to be a severe variety of if any baby has this type of hypospadias, with or without conditional investment, with or without micropenis, it is considered DSD. Then there are battery of tests which you can go. The first tier test, the second tier test. I will not go into it, and you can easily make it out. This slide gives you the approach how to proceed a baby with DSD. So newborn with possible DSD, you have to do carefully. Examine genital examination. If you wish and you are suspecting, you can go for karyotype, FISH, or PCR of an pelvic abdominal exam. And everything depends upon gonads are palpable or not. So again, gonads not palpable. Then you have to look for Mullerian structure. Mullerian structure means you have to look for the ovary, fallopian tube, uterus, upper part of the vagina. If Mullerian structure is absent and gonads are also absent, then you can go for karyotype. Karyotype, if it is 46XY, then it will be labeled as 46XY DSD, no Mullerian structures. If Mullerian structure happens, then you can go for karyotype, and then either it can be 46XX or other. If 46XX, then you can go for 17 hydroxyprogesterone. If it is high, it is indicative of parental hyperplasia. But you should be very careful while interpreting the value of 17 hydroxyprogesterone. In preterm babies, this value of 17 hydroxyprogesterone is more than normal. But if you are suspecting CH, value will be around 80 to 100 times more than normal value. So you will get the value in thousands, not a slightly or borderline range. If your lab is giving value of high progesterone with borderline value, It is not indicative CAH. In CAH, the value of 17 OHP will be sky high. So you should be aware of this. Other things is 17 OHP normal, then it can be 3 beta HSD or for deficiency for oxidase enzyme P450 enzyme oxidative deficiency, placental aromatase deficiency, maternal adrenal ovarian tumors, ovotesticular disease. One very peculiar feature of placental aromatase deficiency is that baby and mother both will be virilized. So what is the role of this placental aromatase during fetal life? So see, if a mother has ovarian tumor, any tumor secreting androgens, first of all, you should know why virilization occurs. Virilization occurs due to intrauterine exposure of the fetus to androgens. This androgen can be of the origin, can be of baby, or can come from mother. Okay, so if this baby is exposed in utero life to androgens, this virilization will occur. And first thing, that if this antenatal or intrauterine exposure to androgen occurs before around 14 weeks, 8 to 14 weeks, if the androgen exposure occurs between 8 to 14 weeks, what will happen? That both the genitalia development and differentiation will be affected. Okay, so this external genitalia will be realized and ambiguous genitalia will appear. But if this androgen exposure after 14 weeks of gestation, this is important. After 14 weeks of gestation, only development will be affected. Differentiation of the genitalia will not be affected. So what will happen? If clitoris is there, it will develop into hypertrophic clitoris. But morphology of clitoris will be maintained after 14 weeks exposure to androgen. Fine. Second thing about maternal aromatase, I was talking. So maternal aromatase can come from any ovarian tumor or any other source which secretes maternal. But if placental aromatase is there, this androgen while crossing the placenta will be deactivated by the placental aromatase enzyme. But if this enzyme is absent in the placenta, what will happen? This androgen, which is of maternal origin, source is mother, will cross the placenta and come to the fetus. It will reach the fetal circulation and it will induce the virilization of the fetus. So, a very common question asked: if mother and fetus both are virilized, then it is placental aromatase deficiency. Fine. So, if karyotype with others like 45X, it is gonadal or some other chromosomal abnormality, ovotesticular DST, mixed gonadal can occur in 46XY. Then again, persistent of Mullin syndrome, Duck syndrome, and gonadal not syndrome. So, coming to gonads palpable, then you have to look for Mullin structures. Again, Mullin structure present, you will go to the same organ. If Mullin structure absent, you can go for karyotype. And karyotype can be 46XX, 47XXY, and 46XY. So if it is 46XX, you will label it as 46XX male. If it is 46XY, you will 46XY DST with no molar structures, or it is 47XXY Klinefelter syndrome. And the Klinefelter syndrome, one universal feature is mental retardation. Actually, the mental retardation increases with the dose of X chromosome. Okay, so if one X chromosome, mental retardation will be with two extra X chromosome, mental retardation will be much higher. So this mental retardation depends upon dose of extra chromosome. So sex chromosome disorder of sex development, 45X Turner syndrome and variants. Phenotypic female with bilateral stick gonads and accompanying somatic abnormalities. Normal Mullin structures absent Wolfenberg. Phenotype female Turner somatic abnormalities. And the single X of maternal origin about 75% of this. Okay. 47XXY. Here one extra X chromosome. The constant feature has small testis, but with histological evidence of impaired spermatogenesis, micropenis, and hypospadias. Here, mixed gonadal degenesis with the unilateral functioning testis, a contralateralistic gonads, all have some mullerian development consisting of an upper vagina, hemiuterus, and fallopian tubule in the side of the aesthetic or absent gonad. On normal side, derivatives of Wolffian structures may present testis 50% mostly invited. 46XX bar 46XY chimeric in both chromosome chimeric means both chromosome composition is present. So in that case, both testis and both testicular in about 50%, ovary in 30%, and testis in 20%. On the side of an ovary, fallopian tube is almost always seen, and on the side of the testis, the vast difference in epididymals are almost always present. On the side with an ovotestis, a vast difference is seen 35% of the time, and the fallopian tube often closed at the fibulated end is seen 65% of the time. A uterus is described in about 6% of present for this. Usually hypoplastic unique for the weight. One more thing I want to highlight, I forgot at the time of development, that this hormone, testosterone, has some local effect and some systemic effect. And it will act ipsilaterally. The local effect of AMH and testosterone is ipsilateral. So suppose one side testis is present and other side testis is absent. In the case of unilateral testis, what will happen? On one side where the testis is present, the Mullerian duct will be inhibited by the Mullerian inhibiting substance. But on the other side where the testis is absent, that side MIS will not be inhibited. So one side where the testis is absent will develop Mullerian structures since testosterone is absent, and other side Mullerian structure will be there. So this is one situation where you will get one side testis, one side differentiation of Wolffian duct, and other side differentiation of Mullerian duct. 46 XY disorder of sex development, disorder of gonadal development, disorder of androgen synthesis or action, and others. Very, very important. So testicular development. So as I told you, this SRY gene is needed for the development of the testis. And development of the testis will depend upon the ipsilateral activity of the testosterone. This due to local effect of testosterone, the ipsilateral gonads will develop. So when SRY gene is absent, the testosterone will be absent, and the differentiation of Wolffian duct into male genital structure will not occur, and this will lead to the dysgenetic testis. Characteristics of this dysgenetic testis are of failure to induce regression of the ipsilateral duct structures, incomplete masculinization of the genitalia, variably abnormal histology, and an increased predisposition towards the development of tumors. I told you these babies are prone for developing gonadal tumors, especially gonadoblastoma. So early gonadectomy is advised. Complete gonadal disease Sware syndrome. It has XY karyotype, presence of Mullerian structures, and since MIS is not secreted, AMH is not secreted, the Mullerian structure will not be inhibited. And as I told you, the default setting is to develop into the female gonads. So Mullerian derivatives will be present. External genitalia will appear as female cells due to the absence of testosterone. I told you again that both the embryo has to develop. The default setting is the embryo wants to develop into female structure in both internally and externally. You have to suppress the expression of Mullerian duct so that this can develop into the male structure. And in case of partial gonadal diseases, clitoromegaly can take place. This SOX9 mutation, SRY like HMG box related genes, very, very important. It is the autosomal mutation. And this SOX9, we don't know exactly how it happens, but it affects the expression of SRY gene. And this SOX9 is not only expressed in the sex chromosomes but autosomes also. So especially the genes which are involved in the collagen synthesis in the bones. These SOX9 gene is involved. So if SOX9 mutation is there, it will cause campomelic dysplasia. So XY gonadal dysgenesis, big, your bony abnormality can is campomelic dysplasia. And actually SOX9 gene products are responsible for collagen type 2 synthesis. So since collagen type 2 synthesis will not take place, it can cause uh, skeletal malformations, bony malformations, that is called campomelic dysplasia. So whenever you are seeing skeletal malformations in a baby with XY gonadal dysgenesis, you can suspect campomelic dysplasia. Other thing is gonadal regression syndrome, and this gonadal regression syndrome can be of two types. Gonads can regress early or late. So we will discuss one by one what is early regression. In early regression, testes will be absent, including the absence of testic gonads, complete absence of both Mullin and Wolfen duct derivatives and female or partially muscular genitalia. Okay, so what happens during the early embryonic life? This testis has disappeared. So testis has disappeared, so it will not secrete testosterone. So what will happen? Either the gonads will be absent or a stick gonad will develop. But by the time the testes have disappeared, it might have secreted anti-Mullerian hormone, and this anti-Mullerian hormone might have suppressed the Mullerian structures. So you are not getting Mullerian structures, derivatives of Mullerian structures. You are not getting the derivatives of Wolffian structures. Either stick gonads or absent gonads can be there. And as I told you, the default setting is to develop into the female genitalia, so clitomegaly and posterior legal fusion can take place in gonadal regression syndrome. Coming to late regression or vanishing testis syndrome, here the testis has vanished due to some reason, somehow late. So here derivatives of Mullerian duct like early regression is absent, but there is a derivatives of Wolffian duct is also present, and the amount of the derivatives of Wolffian duct depends upon the time of regression of the testis. Okay, so the phenotypic feature will be variable. At what point of time this Wolffian duct structure has vanished or testis has vanished? And here can be normal male external genitalia, XY grade 4 testis, or HCG estimation test will be negative because there is no Leydig cells which will respond to the HCG and produce testosterone because the testis has already vanished. But here the testis has vanished late in the course of the disease process. Coming to androgen biosynthetic defects, this slide is very, very important. There are 5 key enzymes involved here. You can see this is a star protein or POR enzyme. Peroxidase oxidoreductase P450 oxidoreductase enzyme is here, star protein. Another is 17 alpha hydroxylase, 3 beta HSD dehydrogenase, 21 hydroxylase, and 11 beta hydroxylase. They are the 5 key enzymes which is involved in the process of this steroid synthesis, steroid synthesis like aldosterone and mineralocorticoid, glucocorticoid, and sex steroids. So, coming one by one, cholesterol uptake into pregnancy. How the cholesterol uptake into the mitochondria takes place. Cholesterol uptake takes place with the help of star protein, and the enzyme involved is P450 oxidoreductase. If this enzyme is absent, all the sex steroids production will be inhibited, and this results in congenital lipoid hyperplasia. Okay. Other important enzyme which is involved in the synthesis is 17 alpha hydroxylase, and this enzyme is not needed for mineralocorticoid synthesis. This is for sex steroids. First 3 hormones enzymes like P450, 17 alpha hydroxylase, and 3 beta and 3 beta hydroxysteroid dehydrogenase are involved, are found in the testis also, and all 5 enzymes are found in the adrenal cortex. So other pregnenolone to 17 hydroxypregnenolone conversion is done by 17 alpha hydroxylase, and this enzyme deficiency can produce deficiency of the sex hormones. If 3 beta HSD is absent, all hormones again will be less by this pathway. But in 3 beta HSD deficiency, 17 OHP won't be raised. Aldosterone, the mineralocorticoid and glucocorticoids, aldosterone and cortisol both will be absent. But sex estrogens can be produced by the peripheral conversion. So it may be normal or slightly less. 21 alpha hydroxylase deficiency, what will happen? That 17 OHP will be raised, but there will be deficiency of aldosterone and cortisol. Fine. So what will happen due to this deficiency? Due to aldosterone deficiency, there will be, and there will be excess of sex estrogens. So here there are two things you can see. One is the horizontal pathway and one is the vertical pathway. Through the vertical pathway, aldosterone and cortisol will be produced. So through the vertical pathway, aldosterone and cortisol will be produced, and horizontal pathway sex estrogens will be produced. If there is disturbance in the vertical pathway, the synthesis of the enzymes of the horizontal pathway, that is sex estrogens, will be high. If there is a disturbance in the horizontal pathway, vertical pathway, enzymes will be more. So this you should keep in mind. So what happens in 2012 alpha hydroxylase deficiency? Here the production of aldosterone and cortisol both will be diminished, and there will be overproduction of sex estrogens. So let us see what will happen due to aldosterone. Aldosterone is the principal mineralocorticoid hormone, and what it will do? So there will be excess of potassium in the blood, hyperkalemia. The role of potassium is to conserve sodium in water. So hyperkalemia will set in, hyponatremia, and water loss from the body. So the baby will be dehydrated. Okay, and maybe may go into shock. Due to cortisol deficiency, there will be hypoglycemia, and cortisol is involved in the hemodynamic stability of the baby. So there will be hemodynamic instability, cardiac failure, and shock will ensue. And due to this overproduction of testosterone, the virilization of the external genitalia will take place. Okay. So coming to star protein deficiency, as I told you, inability to convert cholesterol to pregnenolone, complete deficiency of adrenal and gonadal hormones, that adrenal glands are enlarged with a critically enormous accumulation of lipid, thus the name congenital lipoid adrenal hyperplasia. Males infants are underutilized. P450 oxidoreductase deficiency, CAH plus anti-Baxter deformity, skeletal malformation. Here the craniosynostoses, midface hyperplasia, condyloplasia, stenosis, radioalert stenosis, femoral bowing, and joint contracture can take place. 17 OHP may be elevated and androgen levels are low. Treatment hydrocortisone and fludrocortisone. Treatment hydrocortisone and fludrocortisone should be given. 17 alpha hydroxylase and 1720 lyase deficiency, impaired production of sex estrogens and cortisol, aldosterone normal, high 17 OHP to aldosterone ratio, and hypertension will be a constant feature, and treatment is hydrocortisone plus sex estrogens. In the same way, 3 beta hydroxysteroid adrenal deficiency. Here I told you the clinical picture. I will skip the slide. In this 17 beta hydroxysteroid dehydrogenase deficiency, diminished testosterone synthesis, decreased plasma testosterone and estrogen ratio, if decrease and vast difference may be developed, testis may be presented at puberty must consider express. 5 alpha reductase, this enzyme is involved in the conversion of testosterone into dihydrotestosterone, the more potent form, which is responsible for the development of external genitalia. So in this deficiency, the external genitalia won't be developed. And another is androgen insensitivity syndrome, mutation in the androgen receptor gene, Mullerian structures absent, olefin derivatives rudimentary, testis may be descended to variable extent, female appearing breast development in female body habitus at puberty, XY karyotype, primary amyloid active. Thank you.